years. I'm having looked at these for 3 or 4 years now. It's really great. And we've just got one more master's student, and Megha Sethi is going to talk about the effects of change in flow due to seasonal water abstraction for irrigation on benthic invertebrates of the Manuherikia River system in New Zealand. So Megha's supervisor is Jeremy Potts, another zoology department presenter.
In this picture, it's taken in central Otago in March 2019. More than 50% of the water from the Manuherikia River is diverted into this canal by abstraction to support farms for sheep, cattle, and foods of export quality. But the farmers, with the permit that they have in hand right now, they are not limited to even take 100% of the water from the river. Down at the downstream of this diversion, the depth reduces, the width of the river reduces, the velocity of the flow reduces, leaving the animals in the river, the fish and the invertebrates, to fight for survival during the 5-month period of irrigation. Luckily for the Otago region, in 1991, the New Zealand government passed the Resource Management Act, to comply with which the Otago Regional Council has to renew these licenses and put limits on the amount of water that they could take by 2021. So I, in 2019, went to the site, such 3 diversions of the 213 present on one river, and went upstream and downstream of it to collect invertebrate samples in 3 seasons, when the river was at its lowest, when the irrigation was stopped, and 1 month after the irrigation was stopped. When you feel the back of your neck, you can feel the vertebral column. I am sampling invertebrates which do not have this invertebrate column, such as insects, snails, crustaceans, and earthworms. I take those samples to the microscope in my lab, count and identify each of them, and calculate their densities, their diversity indices, compare them with the variables related to water abstraction, in the hope to give the policy makers a perspective to when they make the policies related to these limits. The invertebrates, they give a very good index to compare the health of the ecosystem. They are the food for the fish such as trouts, tuna, and the native galaxids of this river. Thus, I studied invertebrates. What I am not trying to advocate is that my study will solve the problem of water abstraction in Manuherikia, but just like with any global problem of terrorism, of poverty, it's the collaborative effort of government, of scientists, and platforms such as these where we come together to discuss issues to judiciously and sustainably maintain our freshwater resources.
Thank you, Ray. That was great insight for your project.
Okay, so we forgot to thank you very much. Look, I really I enjoyed your slide. I like the fact that you've got yourself behind that application. And the other thing is that you have explained a few things really well. So again, what an invertebrate is, and then also sometimes you're passionate. You use the word bite, and I love the way that you emphasize the word. Yeah, that's great.
Thank you, Megha. There was a lot of things that I liked in there. I really liked how you brought us into the world of your field study, both through your description of the field work and the slide. And then you took us into the lab as well, so we could really picture the work that was going on in those situations. I liked how you talked about the policy background and then also the implications going forward. And I thought it was very good how you actually said what you're not trying to do. And that was a really good point about the collaborative effort of science and the community and government and everything, and play an important part in its effective solution. So I like that.
Kia ora. I thought I would describe your talk in that you were able to convey science with meaning, and in the passion that you delivered your talk in, you could see that this is more than just say, which is all good to work that's carried out in the lab, that it has meaning to you personally, to the people that you're working with, but also in a global sense as well. And I think that came through really, really well because you were able to add the personal touch to it, as Andrew said, with and Sheila having you in there, that felt like we were there as well. And then also I thought that you had quite a nice political stance. So you weren't afraid to voice that, which can sometimes be hard as a scientist. So well done.